you so much for tuning in. We're pleased to have you join us for the third California Current Acidification Network Ocean Acidification Roundtable discussion of 2018. The title of today's presentation is Risks of Ocean Acidification in the California Current Food Web, Fisheries, and Coastal Economies, Model Projections Informed by Estimates of Species Sensitivity. This series is hosted by the California Current Acidification Network. In short, the series is intended to create a direct dialogue among industry members, natural resource managers, and scientists within the California ecosystem. With awareness of and access to the research and its applications and uses, the participants will be able to collaborate to better understand and adapt to ocean acidification. I'm Teri King, a member of the CCAN Steering Committee and currently the chair, and will be moderating today's session. We also have Shallin Busch from NOAA online, who will not only be presenting, but also running the logistics of the seminar. During the presentation, attendees will be in listen-only mode. You are welcome to type questions related to technical issues or questions for the presenter into the question box at the bottom of the control panel on the right of your screen. We will be monitoring incoming questions and will respond to them or pose them to our speaker after their presentations. We are also recording the session, and we will share the recording on the CCAN website. We're very excited to have Dr. Shallin Busch and Dr. Kristin Marshall speaking with us today. Dr. Shallin Busch is an ecologist with NOAA's Ocean Acidification Program and Northwest Fisheries Science Center in Seattle, Washington. For the Ocean Acidification Program, Shallin staffs the Interagency Working Group on Ocean Acidification, including its new Ocean Acidification Information Exchange, coordinates the program's biological impacts research, and is the point person for the program's activities on the U.S. West Coast. Her research at the Northwest Fisheries Science Center focuses on how ocean acidification may impact North Pacific ecosystems, and she uses laboratory experiments and ecosystem modeling as tools to develop understanding. In 2014, Shallin was stationed at NOAA headquarters, where she helped to draft the National Marine Fisheries Service's Climate Science Strategy. In 2012, she served on the Washington State Blue Ribbon Panel on Ocean Acidification. She currently serves on California's Ocean Acidification and Hypoxia Task Force. Shallin received an undergraduate degree in ecology and evolutionary biology from Princeton University and a doctorate in zoology from the University of Washington. She was a National Research Council postdoctoral fellow at the Northwest Fisheries Science Center. Dr. Kristin Marshall is a research biologist and management strategy evaluation coordinator at the Northwest Fisheries Science Center in Seattle, Washington. Her research focuses on understanding how ecosystems respond to change. Her work seeks to disentangle the signals of human activities, climate variability, and species interactions in ecosystems, and use that understanding to improve natural resource management outcomes. Kristin earned an undergraduate degree from Boston University, a master's in aquatic and fisheries sciences from the University of Washington, and a PhD in ecology from Colorado State University. I will hand the presentation now over to Shallin.
Thanks, Teri, and thanks for organizing today's session. Before I get into my slides, I just want to make sure that folks know that I'm both giving a presentation and running the logistics. So if you have a question and raise your hand and I don't get to it right away, that's why. So just please be patient with us. So I'm really excited to give this talk today with Kristin, and we're going to be talking about two parts of the same project. And the part that I'm going to present on is approaches to estimating sensitivity, species sensitivity to seawater pH. So I was really taken by this image that National Geographic put in their September 2016 issue, where they had an article focusing on the ecological impacts of the marine heat wave that we had in the California Current a couple of years ago. And this beautiful image here kind of goes through some of these food web effects of how species were responding to change. And I think it really resonates with me and in terms of how I'd like to approach the research that I'm doing. And most of my work focuses on, looking at the sensitivity of these little species to change. And I'll go through how we tried to create an ecosystem view of species sensitivity to just one stressor, ocean acidification. So I do most of my work in the laboratory, kind of thinking thinking about the image on the left, where we end up putting individual species or sometimes small collections of species into an experimental treatment system to understand how they might, various life history characters might respond to different ocean conditions. But when people are asking questions about climate change and ocean acidification, they are often asking for this ecosystem view. And it's kind of mind-blowing as an experimentalist to try to think about how you translate what's done in the laboratory to understand physiology and sensitivity to these ecosystem level questions. It can be very mind-blowing. So this is a schematic of the larger project that Kristin and I will be talking today. It was a project led by Isaac Kaplan here at NOAA's Northwest Fisheries Science Center, and included collaborators from University of Washington, JSAO, and CSIRO down in Australia. So what we did under this larger project is develop an understanding of ocean chemistry 50 years in the future. We developed a meta-analysis to understand species sensitivity to ocean acidification, which I'll be talking about. And then Kristin will be talking about the later three parts of the projects, doing the ecosystem modeling and economic impact modeling based on these two projections of the future. One, how the ecosystem is going to respond, and two, how species might respond to those changes. So I'm going to present two papers here. The first one we published, Paul McElhaney is my collaborator on all this work. So Paul and I published this first project in PLOS, and I did highlight in red that this is an open access paper. So anyone who wants to read it can download it, and anyone who wants any of the supplements or databases associated with it can download those for free. So to develop this understanding of species sensitivity of California current species to ocean acidification, we built a database. So it's a global level database looking at any temperate species and how they responded in ocean acidification experiments. And these experiments which expose species to pH conditions expected in the future. So it was current to January 1st, 2015, so it's now getting a little outdated. At the time, we had about 400 papers to choose from that we included, and almost 300 species that are represented in the database. So here are some screen captures of the database that's freely downloadable on PLOS for anyone who's interested. So our focus was the responses in the database. And so just to make clear what I'm talking about here, this is a schematic of how we entered one paper into the database. So this work was by Chris Gobler's lab, published in 2013. There's almost 400 papers like this. And within this one paper, they studied two species, a scallop and a clam. So there were two studies within this paper. And on each experiment, on each species, they measured two responses. And so that's what a response is. It's the data that's collected from an experiment, and then multiple experiments can be included in a paper. What we did is we took an intellectual leap to translate all of these responses into a directional effect of ocean acidification conditions on fitness. So, for example, growth and survival is a relatively easy thing to make the mental leap to fitness on, but something like RNA-DNA ratio, much harder. And that will come up again when I explain how we took this database to really understand and develop this kind of ecosystem level understanding. Just to let people know what folks are studying and publishing on, these are some figures from the paper. On the left side, you can see what % of studies each kind of colloquial group represents in the database. And you can see that there's a lot of work going on oysters and urchins, coccoliths, a lot less work on some of the species that in many ecosystems, especially pelagic ecosystems, are more ecologically important, like krill and copepods. So I point this out just to kind of start highlighting this potential disconnect between the information that we have from scientists and laboratories and the information we need to do ecological modeling. On the right side of the screen, you can see two other kind of metrics, like what people are looking at when they're doing these ocean acidification studies, mostly growth, survival, body composition, photosynthesis, calcification. and then what life stages they're studying. We all kind of know that these early life stages are likely to be more sensitive to environmental conditions, but it turns out that most of the studies are actually on adults. So there's a lot of information from the lab that we have kind of on a global level, but how do we translate that to information that's relevant to species living in the California current? And what we did is we went through a series of rules to give each response a relevance score, where a perfect experiment that would be really relevant to California current species would get a score of 1, and one that would provide very little information on California current species would get a score of closer to 0. And so what we looked at was, where the study was taking place, where the species from the California current are related to the species from the California current. Do the control conditions in the experiment and the experimental conditions in the experiment mimic or are close to mimicking the conditions that are control and expected future in the California current? So it turns out that for many experiments, their future treatment is our current pH conditions. And so that gets you some understanding of why we felt like relevance was really important. And we grouped information at the level of a functional group. So this is a spaghetti diagram of the food web of Puget Sound, not of the whole California current ecosystem, but I put it up here because it's a nice representation of the type of binning we did. Where we're not, for many species, we're not looking at, one species is representing a functional group. It's like a dozens or hundreds of species can represent a functional group within the kind of binning that we did. And when we were developing this study, we thought a lot about how other scientists are trying to put together information to better understand change. And we decided to rely on the IPCC's characterization of confidence when we were developing our methodologies. So their focus here in confidence is looking at the amount of evidence you have and the agreement among those evidence, that evidence. And you'll see how we implemented that in a minute. So here is our version of evidence based on the database. And we said, okay, what is the total amount of information from the database? What are these relevance scores for each functional group that we have in the California current ecosystem model? And you'll see here that we have a lot of information on bivalves, benthic herbivorous grazers, which are things like urchins, but very little information on things like zooplankton, squid, bacteria, a lot of these really important species groups in the ecosystem. And next, we developed an agreement score based on probability theory to measure how much responses agree on how a functional group responds to an increase in CO2. So to put that into clearer words, if we have 2 responses, say on a large zooplankton species, are they both saying a negative effect on fitness under OA conditions? Or are they both saying a positive effect, or is one saying a positive and the other is saying a negative? And so that's how we develop these agreement scores. So we'll go back to bivalves here. You can see that there's not a ton of agreement among the different responses we had in the database. And that could be because some characteristics of species are sensitive to pH conditions and others are not. So there's really legitimate reasons why you get disagreement. I'll show here the Humboldt squid gets perfect agreement, and that's because at this point there was one study in the literature with 2 responses that agreed with each other. So this a high agreement score doesn't necessarily contain a lot of information. So if we would map out the agreement score with the evidence score kind of in this IPCC style, you can see that we have very little information about most of the functional groups that are part of the California current ecosystem. And so I'll just point out here are Humboldt squid with very little evidence, a lot of agreement. And here are benthic filter feeders with lots of evidence, and again, not super agreement. And then importantly, we added a directional score. So we said, okay, what is the net weight of the relevance scores? Is it towards an increase or a decrease in fitness with high CO2? And so you can see here that, some species are going to be a strongly negative directional score, and then other species actually benefit from CO2. So we combined these three things together, evidence, agreement, and direction, to come up with these relative survival scalars. This is the direction and the degree to which we think survival is likely to be affected by increased CO2. And we feel like this relative metric is really the more important, because it's very hard to translate laboratory conditions to what is going to happen in the wild, especially with other stressors, other interactions with species in the ecosystem, and of course, plasticity and adaptation to change. So this is the image that we show most frequently from the paper, where we've kind of transposed this the curve that I just showed you into a sensitivity curve. And I'll just put some characters on this graph to help you better understand who we think is sensitive in a negative way and who we think is sensitive in a positive way. So I've highlighted the top 10 most sensitive functional groups here: urchins, mesozooplankton like copepods, bivalves, pteropods, coralline algae, crabs, shallow benthic filter feeders, which are things like barnacles, crangon shrimp, Dungeness crab, carnivorous infauna, and then deposit feeders here like this potato urchin. And then those species that seem to benefit from high CO2 conditions in the laboratory are macroalgae like kelp, benthic bacteria, gelatinous zooplankton, deep benthic filter feeders, and then seagrass. So many of these are, or not all, but a number of them are photosynthetic. So how do these results compare with other ways of assessing sensitivity to ocean chemistry conditions? And I'm going to show you now a different way we applied the database that I was just talking about to look at a question that seems to have taken hold within the ocean acidification literature, or a hypothesis. And that hypothesis is that species that produce certain types of calcium carbonate are going to be more sensitive to ocean acidification conditions than species who do not produce calcium carbonate, or and those that produce a more soluble version of calcium carbonate will be more sensitive than those that produce a less soluble form of calcium carbonate. And this work is presented in another freely available open access paper in the journal Elementa as part of a special series that some folks here in Seattle put together. And I kind of already talked you through this, but we were focusing on this hypothesis about calcium carbonate and variation in solubility. And just for folks who don't know, shells are actually made up of crystals of calcium carbonate. And what you can see here are some SEM images of what those crystals look like when you look at them at really high resolution. So it's fun to think about, what we typically see as a white shell or a sea urchin spine on a micro level. So as part of this project, we developed a database of all the species in Puget Sound, and we estimated their mineralogy at both the early and the adult life stage. This database includes about 3,000 species and 31 phyla. And like the supplements in the PLOS paper, this Excel table of Puget Sound species is freely downloadable as an Excel file. So what you can see is that 30% of the species in Puget Sound produce calcium carbonate. About half of them don't mineralize at all. 7% form non-calcium carbonate minerals. And then we have this funny category of vertebrate down here. Vertebrate bones are calcium phosphate, not calcium carbonate, but fishes do produce a little otolith, which is calcium carbonate. And so they do have some calcium carbonate in them, but not in the same way that the other species that we classified as calcium carbonate do. So I'm just going to jump straight to the point here with some of our results. What we did is we estimated a survival scalar, like we did in the last database project that I presented. And we said, okay, you know, based on laboratory work, what is the estimated sensitivity of these? And here we're looking at the family level of these species in Puget Sound, crossed with what their mineralogy is, which is on the y-axis. And so if there was this really tight relationship between solubility of calcium carbonate and survival sensitivity, you would see a pattern going from the top left corner to the bottom right corner. And what you see instead is a kind of mess of different data points. So what we drew from this is that species sensitivity estimates based on laboratory research are not correlated with species mineralogy, the way that many people hypothesized. And this is when you look at the sensitivity on a kind of macro ecosystem level. It could be that within the same genus, two species that are very closely related, their mineralogy potentially could influence their sensitivity there. But when you're looking at the class or the family or the order level, we don't see that pattern emerging from the data. And a lot of this, Paul and I believe, is because mineralogy is confounded with phylogeny. So what I'm showing here is two different images. The top one is mineralogy in the early life stage, and the bottom one is mineralogy in the adult life stage. And what you can see is that for each of the calcium carbonate types, you know, what species are producing, they're dominated by a single color, which is the kind of major taxonomic group that they're a part of. And so there are so many other aspects of physiology that are coupled within taxonomy beyond just mineralogy. So we feel like it's really, we're seeing the effect of phylogeny here, not mineralogy. So what are our takeaways? One is that knowledge of species sensitivity to ocean acidification is very uneven among taxonomic groups. And a lot of the places where we have deep knowledge don't have a ton of relevance to ecological modeling. They do have much more relevance to things like aquaculture or to understanding basic biology of sensitivity of species to ocean acidification. But when we're trying to take this ecosystem approach, we find that we have some real glaring gaps in our understanding of species sensitivity. And Paul and I felt like taking this IPCC approach to assess certainty was a really useful framework for synthesizing knowledge on species response. And we hope it'll help people who are doing this IPCC effort incorporate more information on ocean acidification. And then from the second paper, we feel like mineralogy plays a real lesser role to other physiological sensitivities and is confounded by taxonomy, so probably isn't the best way. Mineralogy probably isn't the best way to assess species sensitivity. So that was my last slide. So I'm not sure if there are any questions. If so, I can take one or two, and then I'll pass the presentation off to Kristin.
Shallin, there are no questions that are currently posted, so I'll remind everybody, if you want to ask a question, just type it into the question box. But I definitely have a question for you regarding the database. You indicated that it was, you know, the data work was done up until January 1st of 2015. I find this information fascinating, and I think it would be very helpful. The question is, should this be updated to include the data from, you know, the rest of 2015, 16, and 17? Would it be helpful to the community? And also, at what frequency, if it needs to be updated, should it be reviewed or, you know, reanalyzed?
Yeah, those are really interesting points, and we've gotten this question from a number of people. They say, oh, it'd be great if you could keep it updated. And Paul and I are like, yeah, it's a lot of work. So there are a number of groups around the world that are keeping databases of ocean acidification studies and species sensitivity. Right now, I believe the Ocean Acidification International Coordination Center, which is run out of, I believe, Monaco, is keeping a database that is current. You know, I feel like for Paul and I to continue updating this or to do the bulk of that work to make it updated to 2018, we would want to have another project that would use the database in a novel way. You know, that said, we can give this database, anyone can download it either just on their own and run with it on their own, or talking to us first and then running with it on their own. So we encourage other people to continue to add to the database on their own. And so I will tell folks that the version that we have published as a supplement is kind of a scaled, like it doesn't have a lot of the features in the Access database that help you move around the database easily. And if anyone wants to work with the database and would like those extra tools, get in touch and I can send you a richer version of the database.
And again, this is the PLAWS database.
Yeah.
Correct. Okay, perfect. All right, well, I don't see any other questions. So Kristin?
Great. Can you guys hear me?
Yes, we can.
Perfect. Okay, so I'm Kristin Marshall, and I am going to do sort of part 2 of this and try to take the really excellent work that Shallin just described of doing the synthesis of these different species groups and their vulnerabilities to ocean acidification, and then put that in the context of an ecosystem model and ecosystem model projections that we ran under a scenario of ocean acidification in the future. And this work was part of my postdoc work at the Northwest Fisheries Science Center. And as you'll see, there's a lot of co-authors involved in this, is because a model like this requires a lot of players and a lot of expertise. And so I want to just make sure to acknowledge all of the people that have contributed to this work in many important ways. So this presentation is pretty much follows closely to a paper in Global Change Biology that came out last year. This is not an open access paper, but if anybody wants a copy of it, please send me an email and I'm more than happy to share it if you don't have access to that journal. And so I think, Shallin really sort of gave you the broad overview of this project, and so I just take a minute to try to talk a little bit about the utility of a large complicated ecosystem model and looking at vulnerability to change. And so there's a lot of different things that affect marine ecosystems, from potentially fisheries to climate change, oil spills, lots of different potential stressors going on out there. And it can be really challenging to figure out how all of those different stressors might be affecting individual organisms like this little pteropod here. And then to even understand if they are affecting this little pteropod, what does that mean over the life scale of a pteropod, or over a population of pteropods, or its predators and its prey? And so that's really the main benefit to using a model like this, is to be able to play out how all of these different stressors can interact and what are the cumulative effects of them. And so, this is obviously important, trying to understand the impacts of change, because people rely on marine ecosystems for many different things. So not only individual livelihoods and fishers, depending on harvesting from the marine system, but also, recreational activities, and just people gaining a sense of well-being from being on the water or participating in recreational or commercial fisheries. And so the tool that we're going to be talking about right now is an Atlantis ecosystem model, which is a sort of 3-compartment model that integrates oceanography at the bottom, so a model of climate and ocean conditions, and then uses that to force an ecology submodel and this is really about the food web and all of the marine species that are living in that food web, how they interact with each other, how they interact with the oceanography. And then on top of that is a human impacts model, which is really just our fishing model, our fisheries model. So all of the different fishing activities in the ocean are incorporated here. And so for the purposes of these analyses, what we're doing is using this Atlantis ecosystem model for the West Coast. This model has been sort of under development and going through multiple multiple revisions over the last 10 years. And so we know quite a bit about how it works and how it works on the West Coast. And we're using that model informed by the literature review that Shallin just outlined, so that database of how species respond to OA. And we use that in our ecology submodel. And then we also are using an oceanographic model that predicts pH through the mid-2060s, so through 2063. And then we force our oceanographic model using these future scenarios. And so together, what that allows us to do is ask how individual species and functional groups respond to changes in the ocean over the next 50 years. And the main question that we're really trying to ask here is, what are the components of the ecosystem that are at greatest risk due to ocean acidification? And so we're not trying to make specific predictions about the absolute magnitude of change for an individual species in a single location. This is really a relative exercise. We're interested in trying to look at what are the relative impacts of one functional group versus another over this period of time. And so what I'm going to do is just walk you through the setup of this a little bit, and then go through some results. So I'm going to start with the ecosystem model part and how that's set up for the California current. So here is our map of what our ecosystem model covers, so the area in blue, and the 4 panels around the outside are just zoomed in regions of the coast. But if you look at the panel in the middle, that every number you see on that panel is a different box of our model. And so we have something like 90 boxes where we're explicitly representing food web dynamics up and down the coast. And there's quite a bit of resolution near shore, and that's because that's where most of the things are living, on the shelf and slope. But our model does go all the way out to covers the entire exclusive economic zone, so we can capture some of the big offshore processes as well. So within this, we have all of the species are represented as functional groups. As these are the same functional groups that Shallin was using in her description of sensitivities to ocean acidification. And so we've got 6 primary producer groups and 22 invertebrate groups. And here's just a sample of what those look like. There's 31 different fish groups and 4 cephalopod groups. And these are, have different degrees of resolution depending on sort of how available data is and how commercially or recreationally important they might be. And then we've got 3 seabird groups, 4 shark groups, and 10 marine mammal groups. And again, decisions on sort of how these are lumped is sort of based on similarities in life histories, and then also just what data and information we have to parameterize the model. So that's sort of the overview of the model. This is now what the oceanography looks like, and sort of the contrast in oceanography from 2013 on the left to 2063 on the right. And so what you can see is that using the ROMS regional ocean modeling system setup that our colleague Al Herman runs, from 2013 to 2063, this resulted in about a 0.2 unit change in pH in the summer months. And so what that means, if you don't like to think in pH units, is an increase of about 55% in the acidity of the waters at the surface. And so running this through the ecosystem model had some substantial effects for some groups. And I'm going to just introduce you to sort of how we set up the different scenarios for the different scenarios we ran for the model. And so what we did here, because we didn't want to make everything change at the same time in terms of its sensitivity, because that with a model like this, you can imagine it can be pretty hard to disentangle what's causing what increase or decrease, and just being able to walk through from beginning to end what's changing and what's causing that change is an important part of a model like this. So we broke our scenarios down into 4 different sets of functional groups that we had changing one at a time. And so this shows you the different scenarios. So for benthic non-calcifiers, for example, in that scenario, we had polychaetes, isopods, and cucumbers were all responding to changes in pH, and none of the other groups in the model would respond directly to changes in pH in that scenario. And then the copepod and pteropod scenario, we had copepods and pteropods that were sensitive to pH, but none of the other species were directly sensitive, and so on and so forth. So we had 4 scenarios like that, and then we had a 5th scenario where we made all species sensitive simultaneously, and we call that our cumulative scenario. And so, again, what we were trying to look at was what are the components of the ecosystem that are at greatest risk due to ocean acidification. And so we characterized that by looking at the effects of this change in pH in 2063 with respect to 3 different aspects of the model. So the first is the biomass of the change in biomass of organisms that are directly sensitive to pH. So those groups that I just showed you images of, turning on their sensitivities, what happens to their biomass. The second is the biomass of predators and prey of those species. And so we call those indirect effects. If crabs go down, what happens to the things that eat crabs? And then the last component is looking at potential impacts to fisheries revenue. And for that, we just use present day revenue to sort of, present day revenue to sort of understand the scale of the pH impacts on the different commercially important species. Okay, so now I'm going to go through some results, and I'm going to do this at a couple of different scales. So this is the mean guild response. And so going across the bottom, each one of these labels represents a different guild, so mammals, seabirds, sharks, etc. And a guild is multiple functional groups that fall under that. So this gives you sort of the zoomed out at the ecosystem level. What are the components of the ecosystem that are impacted and which ones aren't? And then the different colors that you see here correspond to the different scenarios that I just introduced. And so each of the colored bars is one at a time. When you turn on the sensitivity of non-calcifying benthos, for example, here, what happens to the marine mammal guild. And so each of the individual scenarios is a different color, and the black is the cumulative scenario when you make them all sensitive. So you can sort of see the parts here. The last piece of this graph that I'll introduce is that you'll see some dotted lines perhaps going out at across plus or minus 0.2. And so 0.2 is the cutoff for what we determine to be sort of a significant impact. And so as you might imagine that in a model like this, there's a lot of uncertainty and there's a lot of noise. And so previous studies that have done this kind of work, looking at different kinds of effects, impose a cutoff around about 0.2 to sort of say things that are less than that are probably noise in the system. So with all of that introduction, this plot is basically telling you that there's sort of mostly small responses overall, and we're looking at the guild level, with the exception of this one blue bar here, and this is the calcifying benthos scenario in the epibenthos guild. And so this is when you turn on the sensitivity for calcifying benthic species, you do see a strong impact on those species directly. But everything else is relatively small at the sort of guild level when you average across functional groups. So this is basically just to say that this is not a doom and gloom story, and so our model is not projecting major dramatic extinctions, for example. It's at the guild level, things seem to be relatively consistent. However, when you start to look at individual functional group responses, you get a more nuanced view. And so here, what I'm showing you is the filled in circles on this plot are identical to the bars that you saw on the plot before. But now you're seeing all of the different functional groups that make up that guild in open circles. And so you can start to get a sense for the variability around the mean response at the guild level. And you see that there's more winners and losers when we look at it this way. And so the strongest responses that you see here are in the demersal fish category over here. And so if we just look at the cumulative bar, for example, what's going on, most of the species that you see that are being strongly negatively affected here are things like flatfishes and some rock fishes. And so these are species that are obviously not directly sensitive to ocean acidification, but their prey species are. And so they're experiencing a negative effect in these pH scenarios because they're losing prey species. A couple of other things to note on this plot, I think, are just the positive. There are a couple of positive effects that we saw over here. And these are the near shore urchins that were showing a positive effect here. They were not parameterized to be directly sensitive to ocean acidification. And so, so they are experiencing a release from predation from some of their predators. Okay, and so now, so that's sort of the direct and indirect biomass effects. And now I'll put it a little bit into context of the economic responses. And so in this plot, what I'm showing you is the baseline revenue on the x-axis. So this is how much was the fishery for a target species worth in 2007, just gross revenue. And then the pH effect, what was the change in its biomass, which is proportional to yield in these scenarios, over that 50-year projection. And so each circle here represents an individual functional group in the open circles, and then what I've done is the filled in circles represent the total revenue for all of the species within these larger categories, and the pH effect is averaged across the species in that group. And so the big take homes here is those, if you look at those yellow dots, all the yellow dots are state managed species. They are the sort of ones from a revenue standpoint make up a large amount of money and have the sort of overall strongest potential pH effects of the groups that we looked at. Oops, I think I just went backwards. Okay, sorry about that. So that's this filled in dot here in yellow. If we look at all of the federally managed groundfish species in blue, you see they overall are worth a little bit less money and are a little bit less, have a little bit lower pH effect score than the state managed ones do. The coastal pelagic species, so things like sardines and anchovies and hake, were less affected in these scenarios. And then the one last piece I'll touch on, as Shallin introduced early on, that this was also linked to some an economics model, and so this is work that Emma Hodgson, who was a PhD student at the School of Fisheries at UW, led as part of her dissertation work. It's currently in review, but she worked with Jerry Leonard, who's an economist at the Northwest Fisheries Science Center, to connect our model outcomes, our model output with an input-output model from an economics input-output model. And basically that allowed us to translate, or allowed her to translate the biological impacts into revenue, income, and employment. And so that's what these maps are showing here. And the cool thing about Emma's work is that she really starts to explore the more fine-scale spatial differences along the West Coast in terms of both the impacts on the biology, but also these economic impacts. Whereas everything previously I've showed you has been sort of coast-wide when you average across the entire West Coast, what are the projected effects. And so what her work here is showing is that there's the strongest economic impacts are happening in the northern ports. And so the darker orangey brown colors that you see are stronger negative effects. And so in the northern ports, you're seeing those stronger negative effects all across the board on revenue, income, and employment. And it's largely driven by the strong reliance on Dungeness crabs. And that's interesting because in contrast to what I'm not showing you, the biological impacts on a spatial map, that those actually sort of overall are stronger in the southern part of the model. So I think this is a really cool piece that's currently in review and hopefully will be out sometime this year. And so I'll just summarize the sort of overall what I hope you can take away from this. And so what we saw overall with our work was that the oceanographic model we were using suggests this 0.2 unit drop in pH during the summer upwelling months. That drop in pH led to direct effects on some of the epibenthic invertebrate species, and those were things like the deeper water urchins, bivalves, shrimps, and crabs. We saw strong indirect effects on the demersal fish, on some of the demersal fish species, some of the rockfish groups, and some of the flatfish groups. We saw indirect effects as well on sharks and then also on Dungeness crab. And so those, again, indirect effects are predator mostly effects on predators of the invertebrate species. Overall, we didn't see a strong effect on the pelagic community, and I think that's partly because of the way our model is set up, and also partly just because we think there may be less of an impact there. And then overall, in the context of the economics, the strongest effects on the fisheries were really on these nearshore state-managed fisheries that tend to focus on invertebrates. Also on the groundfish fishery, but overall that groundfish fishery is worth less than the Dungeness crab fishery. And so that's sort of the overall picture of our paper here. Again, I'm more than happy to circulate it, and with that, I will thank you all and take any questions.
Thank you, Kristin. That was awesome. I really appreciate your perspective and Shallin's perspective and the presentations and the way that you were able to move them together and have them really show us what some literature review, but also some of these other analysis can really do for us in the California current. And I'd like to open the floor to begin our discussion and remind people to type in the questions or comments you have for Shallin or Kristin into the questions box in the control panel.
Well,
and a quick reminder.
While we're waiting, I
Go
have
ahead.
a question for Kristin. This is Shallin. So what is your team doing with these results? Have you all been able to present them to fisheries councils, or are they kind of not ready for management? What is your take on that?
Yeah, good question, Shallin. So Isaac Kaplan, he has shared them as part of the Pacific Fishery Management Council is doing the climate initiative, working on a climate initiative right now, and as part of that, they're doing a series of webinars on climate impacts on the West Coast. So Isaac's presented some of this work as part of one of those webinars. It's also been shared with the Pacific Fishery Management Council through just the Northwest Center's science updates that my division director, Michelle McClure, does. And so I think it's definitely it's more of a, here are the results from the paper stage right now. Not, I don't think that there's been a lot of sort of back and forth dialogue on it quite yet. But I think that we're thinking about what the next steps are just from a research standpoint, and sort of what would we need to do to make this more useful and more targeted at some of those audiences.
I don't see any questions right now, but Kristin, I do have a question for you. Going back to look at the functional group response, it looked like squid, pelagic fish, marine mammals, and seabirds were all pretty tight to the line. And then as you continued through looking at coastal pelagic species and hake were less affected. So that makes sense related to the mammals and the seabirds. I was curious that you had or that you shared, and that perhaps they're not as impacted. Do you think that's related to the impact that the nearshore fishery gets on a regular basis, or what?
I'm sorry, Teri. I think I didn't catch the very last part of your question. So so I got the first part up through the sort of marine mammals and birds, and then
Gotcha.
I don't know.
Okay.
I cut out.
So I'll jump to the bottom line. The nearshore urchin showed a positive, relation. It didn't seem to be as bothered by acidification. Do you have any hypothesis as to why that might be?
Well, so we didn't make it susceptible to acidification in the model. So that's I guess the more specific answer is it wasn't sensitive, and because it wasn't. We sort of made a cutoff of our sort of top 10 functional groups that we thought that had the strongest impact based on Shallin's work. And so I think the reason that nearshore urchin group went up in the model is because a bunch of its predators went down, and so it became more abundant. Whether that's realistic, I'm not sure, and I think maybe Shallin could speak a little bit more to that, because she probably knows more about the details of the literature that supports that nearshore urchin group and their response or lack of response.
Right. Yeah. So I would think that the fact that that group didn't bump up to the most sensitive might have had to do with the agreement score. There's been so much work done on urchins that people have explored metrics that they know are likely to be affected by ocean acidification conditions, and then lots of metrics that they're exploring. So, for example, with responses of transcriptions of genes, or these other really sensitive physiological markers, it's not necessarily a bad thing that you get a lack of response or a kind of stress response to ocean acidification. And so, it could have had to do with that, but I'd have to dig into the literature a little more to see why that didn't pop up.
Okay, great, thank you. So we do have a couple questions. Eliza Geddes is wondering, in your analysis, did you notice any sensitivity differences between bottom fish and pelagic fish?
Well, I'm not sure. Looking at the timestamp on that question, I think it related to my talk. This is Shallin.
Sure.
So we didn't go in and specify the differences between different types of fishes. So Eliza, I welcome you to look in the database to explore that. I would also point you to Chris Chambers, who's at the Northeast Fisheries Science Center. He's been working on a meta-analysis of fish species to ocean acidification, and I know has this type of detailed information. So perhaps you could connect with him. I will note that I put both my email and Kristin's email in the chat box, so you can look at that to get in touch with us. But maybe Kristin, it'd be interesting to see, if you just want to summarize your fish results from the model.
Yeah, I mean, I think that the difference between the bottom fish and the pelagic fish, at least from the ecosystem model response, sort of goes back to what Teri was asking me about, and so I think that the bottom, the benthic fish tend to be more focused on benthic dwelling prey, and so indirectly they seem to be more susceptible to these changes in pH. At least from our modeling perspective, whereas some of the more pelagic species were not seen to have a strong effect, and that was largely because the things that they were eating were not responding strongly to changes in pH. So that's sort of the contrast with the fish from the modeling perspective. And again, those are all indirect effects because we didn't have any fish that were responding directly to changes in pH in the water.
Great. Well, thank you. And we have one more question from Ian Jeffords. It is, what was a parallel look taken at not just pH, but similar effects of reduced aragonite saturation levels? And he sent this in. It could be toward either of you, but my guess is more toward Kristin.
Can you read it to me one more time? Sorry, I'm not seeing it too.
Okay. Was a parallel look taken at not just pH, but similar effects of reduced aragonite saturation levels?
Oh, no, that's a great question, and the answer is that we did not look at different aspects of representing pH. So we only use pH basically because that was sort of the capacity of what this modeling framework could handle at the time. And so certainly you might get different answers if you looked at different ways to represent that, like aragonite saturation states. And so that it is sort of just a limitation of the capacity of this particular model, but a good thing to point out and to know about.
Yeah, and from the synthesis point of view, this is Shallin, we chose pH as our currency because that's what is most frequently reported by people when they publish manuscripts. And so folks working on different taxa tend to kind of target different aspects of carbon chemistry. And not all of them report on aragonite saturation state, and not all of them give you the chemistry parameters that you would need to calculate aragonite saturation state. So for that reason, we also defaulted to pH.
Okay, great. Oops, there's one more. Along the lines of Shallin's question about the North Pacific Fisheries Management Council, have you shared these results with the agencies which regulate state fisheries like Dungeness crabs? And that's from Libby Jewett.
Yeah, that's a great question. And I don't know the answer to that. It's not that I know of, but I will say that, you know, because this was the focus of my postdoc work, I'm working on a lot of other questions now as well. So it could be that Isaac Kaplan, he's really the long-term figurehead of the Atlantis modeling work. He may have reached out to some of the other relevant agencies, but if not, that would be a great thing to do. And so yeah, I'll follow up with him and see what the latest status is.
Wonderful, that would be great. And you know, it's interesting, a couple times in the presentations this afternoon, Dungeness crabs keep coming up, and I think with Emma's work that's going to be coming out and looking at the economics in port and gear and the focus on Dungeness crabs, at least in the northern portion of the California current, that might be an interesting webinar for us to look at all things Dungeness crab as we move forward. So I don't see any more questions that have come in, and so I just want to remind folks that a video recording and PDF of this session will shortly be available on the CCAN website. You can also join the CCAN listserv on the website, where you can also find out about CCAN activities and future webinars. You should also check out the Ocean Acidification Information Exchange and join the CCAN section. I want to thank you for attending the CCAN Ocean Acidification Roundtable. We welcome any feedback, further questions, or suggestions for this webinar series. And it's interesting, somebody pinged me as we were moving through, suggesting one for September. You can submit any input by emailing us at the address listed on the slide. Our next discussion will be on Wednesday, May 16th at 1 p.m. and it's entitled Can Kelp Help? A project to understand if macroalgae can create refuge in a higher CO2 ocean, with Dr. Joth Davis, who is with Bay Water Shellfish and the Puget Sound Restoration Fund, and Dr. Meg Chadsey from the Washington Sea Grant Program. Thanks again to all of our speakers for taking the time to offer their expertise and perspectives about the risks of ocean acidification in the California current. And thank you all for taking the time to join us. This is the end of our session.